All right, well, thank you very much to each of our speakers. Already lots of fascinating insights. So now we will have some time for panel discussion, and maybe I'll start this out with a question for Kayvon. So Kayvon, you pointed out that for KRAS, for mutant KRAS, which, by the way, for those who aren't aware, is of course this is the classical oncogene. It was one of the first human oncogenes to be discovered and has evaded therapeutic intervention for decades, and yet now we might be poised to intervene therapeutically on that. So that's potentially momentous. But you could also say, well, you took the easy road. It's a cysteine, you have a covalent, there are other mutations at various codons. So do we think will this be an outlier? Will cysteine be an outlier? Or do you think are there guiding principles to target RAS that you've learned from targeting the cysteine covalently?
Yeah, I think that's a great question. We definitely treat that as an outlier. That particular drug turns out not to be a great lead to go after the other mutants. So what we've learned from drugging KRAS G12C has not really translated because of the dominance of the covalent bond. But what I think has benefited from that is the motivation to go after the other alleles. And Harold Varmus started the RAS initiative at the NCI, headed by Frank McCormick, that's delving into each of the mutants individually. So I think the G12C taught us it's an outlier, but now we're breaking apart all the other mutants and we're learning particular nuances that have maybe opened up the protein to other mutants that we didn't anticipate. So that's given us a sort of motivation to go after others.
And then how about the general idea that you brought up about inducing a pocket in a protein that otherwise doesn't have a druggable pocket? Is that something that where learnings can come from your work?
Yeah, I think that is a theme that emerges as we think and learn more and use computational tools to understand how drugs bind to proteins and how they adapt. I mean, the drug that Charles didn't get a chance to tell you about that goes outside of the ATP pocket, it binds in such a very flexible pocket, but that's looking great, very good in BCR-ABL. So we do see one-offs, but I hope we get a lot more efforts in those directions. I think they could pay off.
Great. Very exciting. So Nellie, with tumor heterogeneity, you've described a variety of ways of studying it and very elegant incorporation of the Shannon index and other evolutionary models. At the moment, of course, we're not using the full swath of data or even anything close to it in terms of clinical profiling to predict response, et cetera. What do you think, when will the right moment be to take the next step in doing that? And what more information do we need to start doing, or do you think we already should be doing it more?
I think the right moment is now. I think, in breast cancer, for example, in a HER2 positive disease, they have incorporated heterogeneity measure, not exactly the Shannon index, but kind of assessing what frequency of the cancer cells have HER2 amplification. And then based on that, you could, classify the patients into higher or lower risk of treatment. But based on our studies in clinical samples, for example, neoadjuvant studies in breast cancer, how the patient tumor responds to treatment and how the heterogeneity behaves and changes during, for example, the neoadjuvant phase, you could predict what would be the best way to treat that particular patient. So I very much hope that we will incorporate more and more this kind of heterogeneity measures, because we have the data. We have the genetic mutations now tested in more and more tumors. You can just build a heterogeneity index. In many times, we have the immunostaining of the tumors for particular markers that can also be used for heterogeneity measures. And I think, the approach that we have been using, that just try with one drug and highest dose and try to cure a patient, it's not going to work. So we need to be, applying different approaches.
Right. And just stepping back 10,000 feet for a moment, of course, intuitively, we've understood the notion of tumor heterogeneity in billions of cancer cells for a while, and we've known that it can portend resistance, et cetera. But now, of course, with the approaches that you're describing, the resolution, the ability to study it is just unprecedented. So can you share with us, what's something that when you started looking at heterogeneity, you looked and you were just surprised? It was an insight that you didn't expect to see, or what has what was maybe different than what you expected when you started taking a look?
So, I mean, when we started this like many years ago, we initially started with before next-gen sequencing. So, we, looking at like genetic copy number alterations and some of those phenotypic alterations in the cells. And what was striking to me, for example, looking at early stage tumors like DCIS, ductal carcinoma in situ, that you already see a very high degree of heterogeneity in those tumors that, we see most of the changes occurring as in the invasive tumors. And then the question is like, how those tumors evolve, and then how do you select? And in some cases, you could see a subclone in the DCIS that was a minor clone, and then it became the dominant clone in the invasive cancer. So you could see evolution of the tumor happening in front of your eyes, and that was accompanied with some, copy number gain of cyclin D1, and we saw an increase in proliferation. So that was clearly that you could see that this tumor is evolving. So that was one of the very exciting ones. The other one that I found very interesting, genetic heterogeneity, I would say, is the tip of the iceberg, because we can measure more easily with the next-gen sequencing and, all these techniques. But the non-genetic heterogeneity in the tumors, which actually in the evolution slide I emphasized that genetic mutations are selected if they have a phenotypic effect on the cancer cells. So mutation by itself doesn't get selected unless it influences the cancer cells. So those phenotypic alterations, such as changes in the expression of the genes or what kind of antigens they display or something like that, that's even more extensive. And those are not as easy to measure, although nowadays there is this explosion of technologies to looking at multiplex in situ profiling of tumors and really assess genetic, phenotypic, topologic heterogeneity in the tumors and building measures that could incorporate all that into practice. And there is a huge interest in artificial intelligence to use that, because we feel like, we may not have the capacity. We have to incorporate many, many different measures and putting all together to do that. So I'm very excited about application of these measures and AI, particularly in medicine.
Yeah, it definitely seems like the human brain doesn't have enough to just sort of take that in. You need the help of machine learning.
But can I make a point? So your point, Nellie, about phenotypic heterogeneity not being genomically driven, one of the examples I gave towards the end was this concept of lineage plasticity, where an adenocarcinoma turns into a small cell. We've been lately using this technology of single-cell RNA sequencing, and you can see that happening without seeing the histologic evidence. It's not that surprising that it precedes it, but to me the most sort of potentially optimistic way to look at this is it looks like it's reversible depending on how you tweak the environmental conditions. So these are, because they're not genomic, they are epigenetic almost by definition, and there are a lot of drugs that we have in the armamentarium as experimental agents in which we can play with this. So it gets back to this point I think we're both making about we need to try these combinations much earlier.
Yeah. And just one point here, because we have a paper just accepted that epigenetic enzymes, that regulate the chromatin patterns, they seem to be actually regulating cell-to-cell heterogeneity. We saw that with a single-cell RNA-seq, that if you modulate the activity of these enzymes, you can actually make tumors more homogeneous and respond better to treatment.
Oh, that would certainly be an exciting application for epigenetic therapy if that bears out. So actually, let me take the topic of heterogeneity and pivot to Charles, because you made a strong case for the need for combinations, rational combinations, and to get away from the monotherapy-based approach, even if it's very rational targeted therapy. But of course, and then you also commented on the toxicity, which is non-trivial. So it feels like what we need is guiding principles for how can we build these combinations, recognizing the toxicity issues that are unlikely to occur. I mean, undoubtedly will occur when we're stacking various types of targeted therapy. So how have you approached thinking about that? What would you say, where should we begin in terms of guiding principles for rational combinations?
I mean, this is a bit of a soapbox for me. I actually think we're a little too scared of toxicity. And why do I say that? Well, the way a lot of these targeted therapies are developed all through the cycle in academia and biopharma is to go for once a day continuous, pharmacology that allows continuous coverage of the target. But we have evidence in a lot of situations where you don't need to hit the target continuously, and you can still get a comparable therapeutic effect, or you can even get a better therapeutic effect if you give pulsatile high doses in order to induce an apoptotic program, for example. So I would like to see much more creative thinking about not only the pharmacology of these drugs as they get pushed through the drug development system to allow shorter half-life and perhaps pulsatile dosing, but the willingness to use combinations much earlier in a cyclical fashion, much like we give combination chemotherapy in cycles. We have really powerful measurement tools to go beyond just a CAT scan and look at the effect on tumor burden using circulating plasma, DNA, et cetera. And there's a lot of data that would argue that if you can increase the slope at which the tumor burden drops, you really are going to impact long-term outcome. So it's almost like a cultural mindset about being willing to aggressively go to combinations earlier and perhaps use heterogeneity indices, et cetera, per Nelly's talk, to pick the patients who are most likely to benefit from a combination therapy earlier rather than just waiting until they've been through, sequential therapy, and then they're so heterogeneous that the theory's not going to be as promising.
Yeah, and just to
quick plug
please,
for the
yes.
chemistry to add to that. I think one thing we really need is we need many more drugs that are oncogene mutant specific, like the osimertinib you talked about, and IDH mutant specific, and the G12C one, because when they're mutant specific, then when we combine them with a drug that is both mutant and wild type hitting, that won't be the same toxicity as when we combine two drugs that hit both the tumor and the body. So as we get more of those, I think that'll be a big step.
Great. So let me just extend this conversation a little bit to resistance to immunotherapy. And Tony, you made the point that, at least simplistically, if the tumor has T cells that obviously where they're trying to attack the antigen, often that's the context where you get response even to monotherapy. But then you sort of had the figure on the bottom where there are no T cells, and that now begs the question, are there some tumor types where, for whatever reason, there just aren't going to be the T cells or the right T cells, and are we sort of stuck now coming up with more elaborate ways to bring the T cells in, or more or less some patients destined not to benefit from this exciting new advance?
If there's nothing that triggers the T cell receptor of a T cell, none of this will work. And the T cell receptor recognizes something that's abnormal in the cancer from what's in the normal cells. The best example would be mutations that are specific for the cancer. And the more mutations, the more responses. The mutations, as my friend Ton Schumacher says, is like buying tickets to a lottery. The more mutations a cancer has, more likely it is to respond to these therapies, but there's no linear correlation. So there's other things that play there. There's aberrantly presented proteins. There's viral proteins that could be the targets. If those are not there, if that tumor is completely silent, this will not work. But we're seeing that there's something about initiating the response. So the new therapies that we're doing, we're changing a tumor locally by injecting something cholestoxin kind of thing and amplifying it with a PD-1 blockade. It would be like having a pile of wood and nobody has put the first match to start it. We can start that ongoing, and then the immune system will amplify that response. Obviously, we'll never cover everyone. So there is going to be a limit to where we get with these therapies.
Great. I'm going to ask a follow-up question to Tony, but after that, we'll open up the mics to the audience. So if people have questions that have been burning, feel free to come up to the mics, and then we'll give you a chance to ask. But the follow-up question is, if you step back now and think about resistance, I mean, you showed very nicely your diagram of, on the one hand, you could have beta-2 microglobulin mutations so you don't present, and on the other hand, you have an interferon, and it sounded very simple. But we also heard from Lori this morning about the hostile microenvironment that's not Mr. Rogers' neighborhood, but there are a lot of players in that environment. You've got myeloid suppressor cells and Tregs and secreted factors and metabolites. So it feels like it's going to be complicated. It feels like resistance is going to be really complicated. So how do we reconcile the simplicity that you showed with the complexity in the microenvironment and sort of figuring out how you would take the next step of overcoming that resistance?
Obviously, anybody who's gone to an immunology course recognizes that the immune system is complicated, especially with tumor, and we've scared many people for many years with how complicated we made it. But once we have a therapy that works, then we can start understanding it, and we can start simplifying what are the interactions and what could change. But that has to lead us to what do we do next? What is the next steps? And there's undoubtedly many other things that were not in that cartoon that are toning down an immune response. And Lori correctly pointed out to these myeloid suppressor cells that their role is to turn off an immune response. There's Tregs, there's immune suppressive metabolic factors that are not allowing a T cell to start engaging these. We're talking at lunch, are there too many drugs being developed in immuno-oncology and too many combinations? I think no. I think the majority of those agents that are being brought and leading to combinations with these checkpoint inhibitors have years of literature about their immune suppressive role, and we want to get rid of those. Only with testing humans will we know which one is more important. There's no model for the human system, so everything else is just modeling a little window of it.
Right. All right, so I'm going to go to the mics now, and if you could just state your name and institution before asking the question.
Sam Silverstein, Columbia University. It seems to me that the heterogeneity of tumors provides a real opportunity to understand how T-cell immunotherapy actually works. It's unlikely that most tumors have where T-cell therapy is curative, still are heterogeneous, and many of the cells in those tumors either lack MHC or lack the peptide that the T-cells are directed against. What do you all think are the mechanisms for the final cleanup of those rare mutant cells that must be in almost every tumor?
We start these immunotherapies with a lower tumor bulk. In general, we tend to have better responses. So indirectly it tells us that a more evolved tumor, probably with more divergent heterogeneity, will have more innate systems to escape the immune system. If that's a highly immunogenic tumor, like the colon cancers that have microsatellite instability, if we look at those tumors, even before we do immunotherapy, there's clear evidence of something called immunoediting, where the immune system pressure has allowed that cancer, part of it, to change some genes to escape from being recognized. So I think both things are true. It tells us that the earlier we intervene, the better we'll do, and we prevent heterogeneity or tumor growth will be better. But that doesn't mean that we don't have responses in patients with wildly metastatic bulky cancers. And that's probably because there was a T-cells that are possibly polyclonal, recognizing multiple mutational antigens that were just there, ready to go, just blocked by one thing. And those patients do well long term.
So does that exclude a role for NK cells or activated macrophages or all of the other mechanisms that might be brought into play by the activated T cells seeing the dominant antigen?
I fully agree with you that there's many other players there. If a tumor doesn't have MHC molecule expression, NK cells gets rid of it. If I had a cancer and tried to implant it into you, your NK cells would reject my cancer. But we're also seeing cancers that after T-cell immune pressure, they lose MHC expression, either by downregulating HLA or losing genetically beta-2 microglobulin, which is required for HLA expression. And those still grow despite a healthy immune system, despite NK cells there. We see the responses once there's a T-cell response that triggers it all.
Let's go to the other side.
Dr. Kyan Gardiner, applied statistician. This morning I was thinking about the idea of epigenetics, and this afternoon it was addressed a couple of times under Dr. Polyak's presentation, and I'd like to dwell a little bit on that and perhaps query the application of epigenetics in terms of a triggering on-off mechanism that is also affected by environmental factors. How have you handled it within your research? I'm only a statistician and I'd like to learn.
Certainly.
So epigenetics, I mean, I agree with you, it's fascinating because I feel like it's the readout of environmental exposure to our body. And there are, again, like similar to the other areas of research, there has been an explosion of technologies that now allow us to look at epigenetic profiles of the cells in much higher depth. So very briefly, just summarize, what is epigenetics? It's non-genetic alterations in the genome or non-genetic programs. And tumors are very commonly have a perturbed epigenetic program. There are some tumors that actually have very few mutations, for example, some pediatric tumors, or don't even have any clonal mutation in extreme cases of pediatric tumors, and they thought to be completely driven by epigenetic perturbations that basically makes the tumors not follow the usual differentiation programs. And we can look at these, epigenetic alterations such as DNA methylation and chromatin profiles, and many enzymes that are regulating these epigenetic programs are actually mutated in cancer, in many cancer types, like some of the epigenetic modifiers. And there is an enhanced, effort to now actually look at these chromatin profiles and epigenetic changes at the single cell level in tumors. So we're starting to see that there is this, what I alluded, like, even higher degree of heterogeneity, but it's more difficult to kind of classify states because it's almost like a continuum. It's not like you have a mutation or not, but you could, what I say, like, it's not black and white. You have many shades of gray in tumors, so that's why they're not like normal tissues where you have defined states, and that makes it challenging to address in terms of like even describing it. And then that what makes the tumors very, very plastic in a way that they can adapt to the environment. But from a statistical point of view, I think it's very exciting also because we're starting to understand what really regulates these epigenetic programs in tumors, how we can potentially modify them by targeting particular regulators of enzymes. So I think that part of it that we can actually modulate it distinguishes it from the genetic heterogeneity, and I think will be, we need to understand it better, but will be exciting to explore.
Great.
Mark Fishman from Harvard. I have a completely naive question. When you think about evolution, very few people read Darwin's second book, which is The Descent of Man, where he talks about the things he was bewildered by. And one of those was how evolution can work through altruism. Is there any evidence that tumor cells ever sacrifice themselves for the good of the whole?
So I'd like to respond to that, because if you read the literature in the 1980s, this heterogeneity is not a new concept. We knew that. And there are actually articles, very thoughtful articles by Gloria Hepner that exactly discusses this, like cancer cell communities. And the altruistic behavior, the closest I would say, is what we call the non-cell autonomous effect of cancer cells, which I think is actually, we've shown it in our studies, and non-cell autonomous action is, for example, suppressing the immune system. So I think those are very important for driving tumorigenesis, and in a way you can have a minor clone that's only maybe 10% of the tumor, the total tumor, but that could be really what kind of enables the expansion and growth of the other cancer cells. So I think, I don't know if it's altruistic behavior, but in a way, yes, you can have cancer cells that benefiting the whole tumor, and they actually enable heterogeneity. And we argue that identifying these non-cell autonomous drivers will be actually, and targeting them, or these interactions would be one of the best way to try to control the tumor, because those are the ones that feeding the rest.
Mark, I don't know if this is on point, but there is the example of tumor dormancy. It's most dramatic in estrogen receptor positive metastatic breast cancer to bone, where I don't know if the tumor cell is somehow thinking that its own survival is prolonged by going into a dormant state and letting the host survive longer. But there's a lot of examples of that that are hard to understand because of an aggressive cancer that metastasized in the first place.
Go back over here.
I have two practical questions that are somewhat related. The first is, given what we heard earlier in the morning, and we know of substantial disparities in either outcome or prevalence of some cancers, I wonder how are we doing in terms of inclusion of diverse populations in the kinds of studies that you're talking about? So that are we learning about how some of these combination therapies might benefit different populations? That's sort of question one. And in relationship to that, Story Landers began the morning by talking a bit about her personal stories with family members. And I happen to have two close family members who had, in the last 2 years, treatment-resistant cancer. And because I'm at a university, a leading university, I was able to help them identify trials of combination medications, and they ended up both having like miraculous responses. They were in very small, early trials where they had not been tried before. And I couldn't help but thinking, if it weren't, someone in an academic center who could find out about these trials, how are we going to be a world that can kind of quickly translate some of these opportunities to more diverse populations?
Maybe I'll start on that because I thought about this before I left academia and also on the industry side. I think the short answer is we're not doing nearly enough. And you probably know that if you look back at enrollment of diverse populations, for example, African American populations on oncology clinical trials, they were always low to begin with, but you can argue that over the past couple of decades, the rates have even gone down in some context. So what clearly is needed, obviously a lot of this has to do with not just the infrastructure for running clinical trials and increasingly sophisticated clinical trials where one wants to biopsy and then get these elaborate profiles, although that's clearly a big part of it, but it's also the ability to engage patients and have them have the time in their lives to participate in the study. So it's a real issue, and ultimately, part of what's needed is to start where progress can be made. There are sites around the country that are maybe farther along with some of these infrastructures and to forge the right relationships there to get things started. But it is a challenge, and it's a challenge that unfortunately will take a lot of lift from a lot of different stakeholders before it really begins to get better.
And just add one thing. A lot of the eligibility criteria for targeted therapy trials are based on molecular abnormalities in the tumor. And when you look at the databases that lead to the decisions about those eligibilities, it's based on large cohorts of patients who've been sequenced in which underrepresented minorities are severely underrepresented. And then when you then take the next leap, which is that in order to know if you're eligible for such a trial, you have to have had your tumor sequenced, and that's another slice that discriminates against those populations. So this is an awareness in the community. The New York Genome Center, which I have some role in, has just announced a tumor sequencing project called the Polyethnic 1000, in which we're trying to, as a first step, try to populate the databases with tumors from patients who represent these other ethnicities to help, guide which mutations are present there and also clinical trial design.
And one other point on this. Here, there's an opportunity to start even one step earlier than this, which is even for the obvious now well-known, clinically validated, several years in clinical use, basic EGFR positive lung cancer, ALK positive lung cancer. There are even disparities even today in use of those basic genetic tests to assign therapy. So even starting there, because now that will elicit an infrastructure that at least now says, okay, well, we can get the right test, and that test can get reimbursed, and lo and behold, now those tests may open up insight into other kinds of targeted therapies. So it even working with the low-hanging fruit can be a meaningful start.
And my name is Bill Hazard. I'm an academic gerontologist. So I'll take you to up the previous conversation. When do you stop looking for treatable cancers? Is it an age phenomenon? Is it a fertility phenomenon? How can you really embrace enough 92-year-olds to test in the usual way, because they're unlikely to enroll? Again, can you speak to these sort of existential issues?
I'll be happy to take that. So there's a move towards taking away barriers to clinical cancer participation in clinical trials. Friends for Cancer Research, American Society of Clinical Oncology, and now the NCI is advocating to take away some of the exclusion criteria that does not allow elderly people with comorbid conditions to participate in these trials. Most of them, there's no reason to exclude. You had this prostate cancer within 3 years. Why would that patient be excluded? Everything else is fine. You have a particular mutation with getting a drug that's not clear through the kidney, but the creatinine excludes you. All of that is going to be changing, and I hope soon. In the comparative group trials, a group network funded by the National Cancer Institute, we want to systematically get away from those restrictions to participation in clinical trials and make it more inclusive. I've treated patients 90, not 100, but very close with these therapies, with the PD-1 blocking antibodies. Nobody will make me exclude any of them afterwards because they're doing well. So every time we do a treatment, there's risks, and we have to be careful about that, and there has to be certain situations that we have to consider. But we should not systematically exclude anybody.
Well, I think I see Story standing, so that means we're at the top of the hour. So, but. Well, first of all, I'd like to thank the panel for a very exciting discussion. It's been wonderful to see the examples of success, but also to get a chance to dig a little deep on the challenges that remain, which are considerable. But I think what we've heard are glimpses of the directions that will lead to the next time that NAM has a cancer meeting. Hopefully we'll be citing many more examples of success. So thank you.
Thank you very much.